Good morning, everyone, and also online. Today, I have a pleasure to introduce Dr. Jingjing Sun. She is assistant professor at the Department of Pharmaceutical Sciences at the University of Nebraska Medical Center. And she just started there in September in 2023. And before that, she did her undergraduate studies and as well as graduate studies at the Chinese Academy of Sciences. And then before moving to University of Pittsburgh to do her postdoc training there. And then from there, she kind of moving up the rank, become a research assistant professor, where she actually got her own R21 to do some research. And just from talking with her last night, it's kind of amazing to see that, I think Jingjing just kind of did a whole career training of anywhere from chemistry to biophysics to kind of molecular biology and things like that, and how all of her training and experiences really kind of bring her together to what she's doing now with nanoparticle delivery, both chemistry as well as chemical biology. So, and with that, even though she only started in 2023, she already has her R01 starting right off the bat, which is kind of doing some of this nanoparticles. And she has over 40 publications, as well as now like 9 different patents for some of the delivery things that she's doing. And yeah, with that, I would just, give her all the time. Please, welcome.
Thank you, Dr. Nguyen, for your kind introduction. And thank you, everyone, for coming to my presentation, both online and in person. Good morning, everyone. So it's really my honor to be here and share with you my work on targeted drug co-delivery for cancer immunotherapy and synthetic lethal therapy. So in recent years, nanomedicine has made tremendous progress. We've seen a lot of nanoformulations receive regulatory approval. For example, Jinexo, which use POPLA polymeric micelles to deliver paclitaxel. Doxil, which employs liposome to deliver doxorubicin, and Orpatrol, which use lipid nanoparticles for siRNA delivery. And also recently, we know the COVID-19 mRNA vaccine, which use this lipid nanoparticle to deliver. And also, we can also use siRNA conjugated to galactose for delivery siRNA to liver. So these achievements inspire us to further develop new nanomedicine that can work on cancer combination therapy. We all know that compared to single therapy, combination therapy has many advantages. For example, treatment with one drug may change the way the cancer cell respond to another drug. So these two drugs may have synergistic effect in killing tumor cells. Besides, tumor is highly heterogeneous. Some tumor cells may respond to drug A, and some cancer cells may respond to drug B. If we combine these two drugs together, it can further enhance the therapeutic efficacy. So, and compared to free drug combinations, using the nanoparticles like liposome, dendrimers, micelles, or nanogel to deliver these therapeutic agents can further improve the pharmacokinetic and biodistribution profiles. Besides, with rational design, we can allow the simultaneous delivery or programmable delivery of multiple drugs at their optimal dosage to the tumor sites, which can further maximize the therapeutic efficacy while reducing the off-target effect. However, there is limited number of nanomedicine that has been used in clinical combination therapy because the current nanocarriers has very low co-delivery efficacy due to their different physical chemical properties of different drugs. So in addition, it is unclear about how the nanocarrier interact with the drug and how the nanomedicine interact with the biological system. Understand these interactions will be very helpful for future rational design of nanocarrier. With these challenges in mind, our lab is collaborating with other research groups to identify new targets through bioinformatics and molecular biology. And we are also investigating developing new combination strategies, including the use of small molecular drugs, DNA, RNA, genome editing system, and PROTAC system. So to co-deliver this multiple therapeutic agents, we employed chemical synthesis tools to develop polymeric nanocarriers or lipid nanoparticles. And through computer modeling, we will investigate the interactions between the therapeutic agents and the nanocarriers and understand why the nanomedicine can selectively go to the tumor. And we will also investigate how the nanomedicine modulates tumor immune microenvironment and also how the nanomedicine targets signaling pathways in the tumor cells. Today's presentation will cover 3 parts. In the first part, I will talk about a nanocarrier based on gemcitabine conjugated polymer to deliver CCR2 antagonists for pancreatic cancer immunotherapy. And in the second part, I will talk about using the azacitidine-based nanocarrier to deliver PARP inhibitor for enhanced synthetic lethal therapy. And in the last part, I will briefly talk about some new nanocarriers that for efficient delivery of siRNA and mRNA. As we all know, pancreatic cancer is very difficult to treat because of the dense plastic stroma. This stroma not only restricts the nanomedicine penetrate deep into the tumor core, but also limit the infiltration of effector T cells. As a result, so the pancreatic cancer is considered non-immunogenic. So the immune checkpoint blockade therapy, which is effective for many tumors, is less effective here. So it is crucial to develop new nanomedicine that can penetrate deeply into the pancreatic tumor core, but at the same time can modulate tumor immune microenvironment. It has been reported that particle size play a role in tumor penetrating, particularly for pancreatic cancer. There is a paper published in Nature Nanotechnology. They found that nanoparticles smaller than 30 nanometers shows better penetration compared to larger particles like 50 or 75 or 100 nanometers. So, but when reducing the particle size, the drug loading capacity and formulation stability also decreased. So to address this issue, we developed an ultra-small nanocarrier based on gemcitabine conjugated polymer. Here, we conjugated gemcitabine to a specific polymer backbone, PVD, to give this PGEM, we call it PGEM. So surprisingly that after gemcitabine conjugation to this PVD polymer backbone, the particle size was significantly decreased from 160 nanometers to 13 nanometers. And this small size is beneficial for tumor penetration. More importantly, the PGEM overcomes the limitation of a small-sized nanoparticle in loading drugs. It can load a wide variety of hydrophobic drugs with very high drug loading capacity. Here you can see it can load paclitaxel, IDO inhibitor, NLG999, doxorubicin, curcumin, and most of them showed high drug loading capacity, like 24%. In general, the nanocarrier can only load less than 10% drug inside the nanoparticle. So compared to the larger PVD nanoparticles, smaller PGEM nanoparticles showed higher accumulation in tumors, both right and left tumor in the patient-derived xenograft model. And also here in orthotopic KPC, pancreatic tumor, which contains a lot of stroma, we can see the PGM nanoparticles mainly accumulate in the tumor rather than in other normal organs. So then we got this tumor and frozen section and stand CD31 antibody for the blood vessels. Here you can see for this rhodamine signal comes from the nanoparticle. Here you can see for larger PVD nanoparticles, it mainly locate around or near to the blood vessels. But for smaller PGM, it showed widespread signals, which means it has better tumor penetration capability. We treated the tumor-bearing mice with saline, free gemcitabine, and PGM, and then harvest the tumor for RNA sequencing analysis. Through analysis, we found that PGM nanoparticles downregulated several gene sets involved in pancreatic cancer pathogenesis. That's because we conjugate gemcitabine into our nanoparticles. And then we also found that PGM treatment led to increased cytosolic DNA sensing pathway and interferon alpha pathway. So if you look at this diagram, you will see these two pathways are involved in C-gas STING type 1 interferon signaling pathway. This pathway can be activated in both tumor cells and dendritic cells. In dining tumor cells, the DNA damage lead to the double-strand DNA, which can be recognized by C-gas. And then this can further catalyze the formation of C-gap and activate STING, which promotes the release of type 1 interferon. This is very important for dendritic cell maturation. And the STING pathway activated in dendritic cells can be initiated by exosome release, engulfment, and gap junction. And the dendritic cells will migrate towards the tumor draining lymph node and the cross-prime T cells, which traffic through the blood vessel to conduct anti-tumor killing. So based on this information, we hypothesized that this PGM nanocarrier itself may activate C-gas STING type 1 interferon pathway. To confirm our hypothesis, we evaluated the presence of cytosolic DNA by using the pancograin staining and then evaluated its co-localization with C-gas. Here you can see PGM treatment increase the percentage of the cytosolic DNA and its co-localization with the C-gas. Western blot shows you that PGM treatment increased the expression of phosphorylated STING and phosphorylated IF3, which is the downstream marker of STING activation. In vivo studies, so we treated the tumor-bearing mice with free gemcitabine and PGM, and then harvest tumor for flow cytometry analysis. Here you can see PGM treatment significantly increased expression of phosphorylated RF3 and phosphorylated TPK1 in both tumor cells and dendritic cells. So this, like phosphorylated RF3, the TPK1 is also downstream marker of STING activation. So this study suggests that PGM treatment can activate C-gas STING signaling pathway, both in vitro and in vivo. We also found that PGM treatment increased the expression of interferon beta in both cultured cells and harvest tumor tissues. So, which means PGM led to enhanced expression of type 1 interferon genes. But we also found some bad news here. PGM treatment also increased the expression of CCL2 and CCL7. This was further confirmed by our qPCR. So here you can see PGM treatment increased the expression of CCL2 and CCL7, both in vitro and in vivo. So many of you may already know that CCL2 and CCL7 are immunosuppressive chemokines. They are major tumor-derived factors responsible for recruiting immunosuppressive immune cells like tumor-associated macrophages and MDSC, so which promote tumor progression and metastasis. It can also promote the production of cancer-associated fibroblasts, and they may be involved in tumor resistance to various treatments. So we would like to combine PGM nanocarriers with inhibition of CCL2 and CCL7. Luckily, CCL2 and CCL7 share the same receptor, CCR2, so we can only just load CCR2 antagonists into the PGM nanocarriers to address the negative feed mechanism. So through molecular dynamic simulations, we screened a lot of CCR2 antagonists and found that PF6309 is a good CCR2 antagonist formulated into PGM nanocarriers. And this formulation shows excellent formulation stability at room temperature and at 4 degrees. And also they showed very small particle size around 15 nanometers. And compared to free drug combination of CCR2 antagonists with gemcitabine and PGM nanocarriers, loading PF into PGM nanoparticles significantly increased the therapeutic efficacy. And in the lung metastasis model of pancreatic cancer, we found that this new nanomedicine led to fewer tumor nodules in the lung tissues, suggesting better anti-metastasis effects. So we also evaluated the changes in the tumor immune microenvironment after various treatments. Here you can see PGM nanocarrier alone led to increased percentage of MDSC and tumor-associated macrophages in the tumor tissues. This may be due to the higher expression of CCL2 and CCL7 induced by PGM, which recruit the MDSC and TM in the tumor tissues. But when we loading the CCR2 antagonist PF into PGM nanoparticles, it significantly decreased the percentage of MDSC and TM. So this means loading CCR2 antagonists into the nanoparticle can reverse MDSC and TM immunosuppression induced by the PGM. We also found this new nanomedicine increased the percentage of NK cells and granzyme B and interferon gamma positive NK cells, which are functional NK cells. Also, this new formulation increased the percentage of CD8 T cells and functional CD8 T cells. So this means this new nanomedicine can activate both innate and adaptive immune response. Here you can see PGM-PF nanoparticles also increase the expression of PD-1 on NK cells and CD8 T cells. So this provides us a new opportunity to further combine with PD-1 antibody. So here you can see compared to free antibody PD-1 treatment or our nanoparticles, combination of these two can further enhance the therapeutic efficacy and with very low tumor weight at the end of experiment. And one of the tumor totally disappeared after 3 treatments. So to summarize this part, we developed a PGM small size nanoparticle. So it shows selective tumor accumulation and efficient tumor penetration. In another study, we use this PGM to load paclitaxel for pancreatic cancer therapy. And this these nanoparticles receive orphan designation from EMA and FDA, and now scale up for physical one clinical trials, maybe happen later this year in Australia. And then here we evaluated the mechanism of the PGM. We found this PGM nanocarrier can activate STING and induce innate and adaptive anti-tumor immune response. Besides, it can induce the expression of CCL2 and CCL7, which recruit immunosuppressive MDSC and TM. To address that, we loaded CCR2 antagonist, PF, into the PGM nanocarriers, which can reverse the MDSC and TM mediated immunosuppression and further enhance the therapeutic efficacy. Besides, we combine the nanoparticle with anti-PD-1 and found that the nanoparticle can sensitize pancreatic cancer to anti-PD-1 therapy. In the second part, I will talk about using the piezo nanoparticle to deliver PARP inhibitor. So still we use same polymer backbone, and we found when we replace gemcitabine with 5-azacitidine, this new particle also shows small particle size with high drug loading capacity for a wide variety of drugs. So 5-azacitidine is an inhibitor of DNA mass transfers, and which overexpress in tumor cells. Compared to normal cells, tumor cells have abnormal demethylation patterns with hypermethylation in DNA promoter region, leading to transcriptional silencing of many tumor suppressor genes. So 5-azacitidine can incorporate into the replicating DNA and then cross-link DMT to create DMT, DNA cytotoxic complex, while simultaneously degrade DMT. So this is very interesting drug. At low dose, it serves as epigenetic modifier through inhibiting demethylation, but at higher dose, it mainly shows cytotoxic effect through inhibiting DNA synthesis. So first, we also check the biodistribution of this nanoparticle. Here in orthotopic lung cancer model, these bioluminescence signals indicating the tumor cells in the lung. And these DRR fluorescence signals comes from the nanocarriers. Here you can see for larger PVD, it mainly accumulated in the liver. And for smaller piezo, it mainly accumulated in the orthotopic lung tumors. So this is quantification data. You can see it mainly shows higher signals in the lung tumors than in other organs. So we use these piezo nanocarriers to load PARP inhibitor. We chose PARP inhibitor because we know when we use PARP inhibitors, the PARP can trapped to the DNA break sites and lead to the double-strand breaks accumulation during DNA replication. And in HR proficient tumor types, the homologous recombination can repair those DNA breaks. But in HR deficient tumors, such as the mutation or deficiency in BRCA1, BRCA2, or RAFE2-1, the homologous recombination will no longer occur and the cell will be killed through synthetic lethality. So here we know PARP inhibitor mainly works for the HR deficient tumors, but unfortunately, most tumor HR proficient. So to expand the application of PARP inhibitor to HR proficient tumors, so we deliver the PARP inhibitor using the piezo nanoparticles, because here, as you see, this RNA sequencing analysis shows you compared to BMN, which is a very potent PARP inhibitor. Using the piezo to deliver PMN can reduce the DNA repair signaling pathway, which was further confirmed by RAD51 nuclear foci. Here in the HR proficient non-small cell lung cancer model, we can see the BMN treatment led to the increased signals of red fig nuclear foci, which means maybe after BMN treatments increase the HR functionality. That's why PARP inhibitor doesn't work that well in HR proficient tumor cells. But when we use PISA to load BMN, the red FT1 nuclear foci were significantly decreased. Then we evaluated the gamma H2X levels, which is a marker of double-strand break. So here you can see in this HR proficient non-small cell lung cancer, free azacitidine or BMN could not induce the DNA damage, but when combination of these two drugs or loading BMN into PISA nanoparticles, it significantly enhanced the DNA damage. So here shows you the in vivo data. So free azacitidine or BMN, this red line and this orange line and this green line could not inhibit tumor growth. But when we combine these two free drugs together, it can increase the therapeutic efficacy. And also compared to free azacitidine, conjugation azacitidine to our polymer significantly increase the therapeutic efficacy, which may be due to the increased availability of azacitidine, because azacitidine is so easy to degrade in vivo, like just several minutes, it can be degraded. So here we can see compared to free drug combination, loading BMN into PISA further enhanced the therapeutic efficacy. And to our surprise, when we further decrease the dosage to 0.25 milligram per kg, it shows better therapeutic efficacy. This may be due to like azacitidine works through the DNA methylation inhibition at low dose, but maybe also due to like immune response, enhanced immune response. We are further investigating this mechanism behind this. But anyway, so this new nanomedicine works at very low dose, which shows great potential for clinical translation. Then we found this is an ultra small nano carrier platform that apply to many nucleoside analog drugs. Also like the FCR, which is 5FU prodrug. So this nanocarrier also can load a wide variety of hydrophobic drugs with very high drug loading capacity, and also show the selective tumor accumulation. This shows you the colon cancer tumor models, and you can see it did mainly accumulated in the tumor, and you can barely see signals in other organs. So this platform can be used for co-delivery of multiple water-soluble and water-insoluble drugs together for various combination therapy in various cancer models. But there was a question for us how these small nanoparticles interact with biological system and mediate the effective tumor accumulation and penetration. To answer these questions, first, we evaluated the cellular uptake. Here we can see compared to PVD, the smaller PISA was more effective in internalization in the Lewis lung cancer cells. And we also, by using different endocytosis inhibitor, we found that both PVD and PISA were taken up by the Lewis lung cancer cells through class-mediated endocytosis. So the endocytosis pathway didn't play a role in the difference of the PISA in tumor penetration. So in the 3D tumor sphere model, we can see compared to PVD, smaller PISA was more effective in penetrating through the 3D tumor spheres. When we use the endocytosis inhibitor, chlorpromazine, the tumor penetration capability was decreased. This indicates that endocytosis may play a role in tumor penetration. So to further confirm it, we did another transwell study with multi-layer tumor cells in the upper layer, upper channel, and then we incubated the fluorescence labeled nanoparticles and evaluated the penetration. And here you can see PISA was more effective in penetrating through multi-layer tumors compared to PVD. When we use endocytosis inhibitor, the penetration capability was significantly decreased, which further confirmed that endocytosis play a very important role in the tumor penetration for the PISA. So initially, we thought the PISA nanoparticle penetrate tumor through the paracellular transport due to the small size effect of PISA. But those results told us maybe transcytosis play a role here. So, but we don't have any targeting ligand on the nanoparticles. So what mediated this transcytosis? Recently, there is a paper reported that actually most nanoparticle, like 90% nanoparticle after administration, will form a protein corona on the surface of nanoparticles. This protein corona give the nanoparticle new identity, which help them interaction with immune systems and interaction with target cells. So we were thinking whether our PISA nanoparticles also coated with some protein corona that mediated transcytosis. So we incubated PISA and PVD separately with serum and isolated nanoparticles for proteomics analysis. And we found the fibronectin was more enriched in the protein corona of PISA than the PVD. The fibronectin contains the RGD sequence, which can interact with integrin alpha, which overexpressed on the tumor cells. So we think maybe fibronectin mediated on the surface of the PISA nanoparticle mediated this transcytosis. To confirm it, so we did another transwell study under different conditions. Here, you can see in the absence of serum or fibronectin, both PVD and PISA nanoparticles shows very low penetration capability. When we add the fibronectin or serum, the penetration ability of PISA was significantly increased. But when we incubate with the antibody against integrin, then the penetration ability was significantly suppressed, which indicates the important role of fibronectin and integrin receptor. We further knock out the RTGA5, which is integrin alpha 5, which is a receptor of fibronectin. So, and we found that knockout RTGA5 led to a drastic decrease in the transcytosis for the PISA nanoparticles, but not for PVD nanoparticles. And in another tumor sphere studies, we found that the penetration ability of azacitidine was higher than PVD. And when we depriving of serum in the medium, the penetration ability was decreased. When we add fibronectin again in the medium, it's the penetration ability of PISA was partially recovered. So that means fibronectin is very important for penetration of the PISA. We also, in the RTGA5 knockout established tumor sphere, we found that penetration capability of PISA was significantly suppressed. So this means RTGA5 receptor is also very important. To evaluate the in vivo effect, we inoculated RTGA5 wild type and knockout tumors separately onto the mice, and then evaluated the tumor penetration ability. And here you can see PISA nanoparticle shows effective tumor penetration in wild type tumors, but significant decreased signals in RTGA5 knockout tumors. So these results suggest that the PISA nanoparticles has the fibronectin-enriched protein, which mediated the transcytosis through interaction with RTGA5 on the tumor cells. So to summarize this part, we developed a new nanoparticle that combines BMN and azacitidine together to inhibit both PARP and DMT, which enhance synthetic lethality in HR proficient tumor types like non-small cell lung cancer. And we found that compared to larger particles, smaller PISA has more fibronectin in the protein corona of the surface, so which mediated the transcytosis and effective penetration through interaction with RTGA5 on the tumor cells. But because compared to PVD, the size of PISA is different. Also, the surface chemistry is different. So in our next work, we want to understand how the particle size and the surface chemistry influence protein corona formation and the biodistribution profile. Also, we want to develop some new protein corona coated polymeric nanocarriers for enhanced tissue targeting. So in the last several minutes, I'll briefly talk about some new nanocarriers that we developed for efficient delivery of siRNA or co-delivery with drugs, and also efficient delivery of mRNA. So we know there are many targets that lack small molecule inhibitors. So for these undruggable targets, siRNA knockdown is a promising strategy, but we cannot inject siRNA directly to the body because it's easy to degrade. So there are many nanocarriers developed for the delivery of siRNA, but most of them primarily accumulate in the liver. So there is strong need to further develop nanocarriers that can deliver siRNA to the tumor, or together with drugs to the tumor. So here we developed two polymers. We found that at 20:1 ratio, these two polymers shows very high luciferase knockdown efficiency, which is comparable to the positive control RMX in pancreatic cancer cell lines. And this shows you the biodistribution data. Polymer 2 here mainly accumulated in the liver. And for polymer 1, in addition to deliver the siRNA to the liver and kidney, it also delivers siRNA to the tumor. This is semi-quantification data. You can see the polymer 1 can deliver more siRNA to the tumor than other organs, but we are currently further optimizing this system. And this system also, another advantage is it can co-load other hydrophobic drugs like PARP inhibitor. So in addition, we developed some new nanocarriers that can efficient delivery of messenger RNA. Here, we developed 4 different lipid nanoparticles, and we use ALC 0315 as a benchmark because this has been used by Pfizer for mRNA vaccine, COVID-19 mRNA vaccine delivery. So here you can see in 41 breast cancer cell lines, the nanoparticle 2 and 3 shows higher transfection efficiency than the benchmark. And in the hepatitis cells, all these 4 nanoparticles shows higher, like 3-fold higher than the benchmark in transfection of luciferase mRNA. So currently we are evaluating the tissue biodistribution of these nanoparticles and then decide what kind of target we want to target mRNA we want to be loaded in. Here, I would like to take this opportunity to thank all my lab members and my collaborators at University of Pittsburgh and UMC. Also very grateful for all the ground support, and also thank you all for your kind attention. I'll be happy to take any questions.
I have microphones for anyone who has a question.
So I really appreciated the part of your presentation where you uncovered clathrin-mediated endocytosis via the alpha-5 integrin as a mechanism for uptake. So how does that mechanism explain the tumor-specific homing? Is it just a simple matter of there being more alpha-5 integrin on tumor cells relative to normal cells?
Yeah, I think for our study, these PIs are nanoparticle because of small size should be both EPR effect and targeting ligand-mediated transcytosis. So because tumor cell compared to normal organs, tumor blood vessels is kind of leaky. So the smaller nanoparticle can penetrate from blood vessels outside to the tumor tissues. Besides, the interferon alpha-5 is also overexpressed in tumor endothelial cells. So this may also mediate some transcytosis through the through the tumor endothelial cells to the tumor tissues.
And do you see that tumor-specific cell entry using in vitro models? So if you were to use like a non-cancer cell type or a non-transformed cell type, do your nanoparticles still get in those just fine?
In vitro models, we just use this tumor sphere or transwell study. Actually, I didn't put another data here. Like we did transwell with the endothelial cells and found this can also mediate transcytosis in the endothelial cells. Probably can answer the questions why it can go to the tumor tissues.
Nice talk. Two questions. The first one is based on your current data, is that will conjugate drug affect biodistribution or the intensity of the tumor targeting accumulation?
Yeah, so that's great question. So yeah, we found that conjugation of drugs like nucleoside analog, no matter it's gemcitabine, azacitidine, or other nucleoside analog, the particle size significantly decreased. But we don't know because the surface chemistry also changed. So we would like to know maybe after this is our next goal. So we will evaluate different chemistry here and also different particle size, how these affect the protein corona formation and the biodistribution.
Okay, next question is all your tumor distribution. The tumor model are xenograft or autotopic or both?
Yeah, some here are sub-Q model, some are xenograft, like a patient-derived xenograft model, and some are also topic model.
So is there any difference between autotopic and xenograft?
Here you can see, I don't see much difference. Here, this is also topic lung cancer model. So you can see it mainly accumulated in the also topic lung tumor. And also another model is pancreatic cancer model. Yeah, this one. So this is also topic KPC, pancreatic tumor, which contains a lot of stroma. So also it's mainly accumulated in the tumor than other organs.
All right, we've got a question online from Doug Yee. A follow-up on Scott's question, the first question. Do you have data on improved therapeutic ratio in the animal system, especially regarding PARP inhibitors and hematopoietic systems?
This question is about the therapeutic ratio, right, of the nanoparticle?
Do you have data on improved therapeutic ratio in the animal system?
So far, we just use the fixed ratio of these two drugs, like azacitidine and PARP inhibitor. This is like a 1-to-1 ratio. We did a preliminary study to change the ratio a little bit, and didn't find significant difference. But these changes, small changes. So in our next goals, actually, we want to further expand the changes of the two ratios and want to identify the optimal ratio of the azacitidine and PARP inhibitor in these nanoparticles for enhanced therapeutic efficacy.
Okay, he follows up with just the quick question, is there less myelosuppression with the particles?
Oh, we didn't do that study, so I cannot answer that question, but that's a great question. We should investigate. Yeah, thank you.
All right, are there any more questions in the room? All right, let's say thank you one more time for a great seminar.
Thank you, everyone.